Everyone, my name's Stephen Locke, and today I'll be talking about how you could use CE to analyze N glycans coming from proteins. Just give me a minute or two and I'll switch my camera off and go to sharing my slide deck with everybody. So today, my presentation is entitled Discover how CE can be used for N glycan analysis. And really, I'll just go through the summary of what I'll cover today. I'll cover the importance of glycans in biopharmaceutical analysis. I'll look at the background to HILIC analysis of N glycans, and then try and compare how capillary gel electrophoresis has been used for glycan analysis compared to HILIC, and go through the theory of capillary gel electrophoresis and how it can be used for glycan analysis. At the end of the presentation, I will just summarize the content today for everybody on the call. So I think really it's always start put an analytical method or analytical problem into context. So I'll start off by looking at the importance of glycans in the biopharmaceutical analysis and understand where glycan testing is used. This next slide shows just a schematic of what happens in the manufacturing of a therapeutic protein like a monoclonal antibody or a plasmid, or a fusion protein or a bispecific. And here you get some ideas how you would manufacture, say, a monoclonal antibody. I think really glycan analysis really starts to kick in when you're looking at understanding your clone or your cell line, which is being used to produce your therapeutic protein. The cells used actually influence the glycosylation of the protein that's being produced. Often you'll find, if you use a CHO line, that this will generate N glycans or the addition of glycans on the protein itself at things like ASN amino acid residues. And glycosylation has some biological significance. It really impacts the activity, the physiochemical properties, ADCC and CDC functions. And as I mentioned, it's really important to do analytical characterization in clone selection and product characterization, as well as lot release. And this is where N glycan analysis really takes place. One of the major sources of glycosylation variety or variance is around the ASN 297 site of a monoclonal antibody. And this variability often depends, as I mentioned previously, on the host cell line and the expression conditions which are used to generate the therapeutic protein. There's quite a lot of structural diversity that can happen. You can have fucose, galactose, sialic acids, GalNAc, as well as mannose type N glycans. So it can be a complex question to answer. As I mentioned, modifications in N glycan processing can have some specific roles on cellular function. And if you look at addition of fucose can impact ADCC activity, GalNAc can impact ADCC and CDC activity, sialic acids can cause inflammation, can impact the half-life of your protein drug, and also can impact ADCC activity, and mannose can also impact ADCC activity as well. So you really try to impart quality by design in understanding the sugar function relationship, i.e. the impact of core, for example, impact of core fucosylation on the ADCC response. So really the selection of cell lines with appropriate glycosylation machinery is important to get the functional relationship for your target biopharmaceutical. And you need to use proper analytical tools to ensure the product you produce is consistent and possesses the correct glycans to enable the function you desire. Now, so the challenge really in this space is that glycans are complex with diversified structures, and there's no chromophore fluorophore groups, so they are mostly not charged, and this really impacts the ability to detect and separate them. So often you have to add a chromophore fluorophore group to your glycan, and then you use separation techniques like HILIC or hydrophobic interaction chromatography, or as I started introducing my talk, capillary gel electrophoresis or CE, to do the analysis of labeled glycans. So some of the background to HILIC analysis of N glycans. Now I'm talking about labeled glycans, but firstly what you need to do is actually remove the glycans and capture them before you analyze them. So typically you have to digest the glycans off the proteins before you can analyze them. And analysis, for example, with HILIC uses a liquid-liquid partitioning chromatography, where analytes, the glycans, elute in an order of increasing polarity. Now, what you'll often find with HILIC, of course, if people who know HILIC, it's the reverse elution order compared to reverse phase LC. And most LC column manufacturers, for example, Phenomenex, will produce HILIC columns. And most current HILIC columns use small particle in their columns and higher pressures because these produce better resolution and can also be applied to make the analysis faster. And after you run the analysis and you get your chromatogram, then your glycans are identified by the GU value by comparison to a reference standard. And GU stands for glycosylation unit. So as I mentioned, in order to do this analysis, you first have to cleave off your glycans, your N glycans from your protein, your monoclonal antibody, and the common enzyme used in this space is PNGASF. So you digest your protein, then you label it, and one of the common labelers, which has been used historically in the past, is 2AB or 2-aminobenzamide. This adds something which can be detected, a fluorophore to the analysis, to the glycan, and then you clean up to remove unused reagent, and then you analyze the sample. And you can see here sort of a typical 40-minute runtime where you're picking up 2AB derivatized N glycans using a HILIC separation on the right-hand side of the slide. This actual 2AB labeling is actually quite a lengthy process. I've just taken off an example of this type of labeling reaction from the website. This is actually taken from an Agilent user manual, and this can say the shift-based process of labeling can take a few hours to do, so it's quite a lengthy process. So there's often a requirement to shift this process to quicker types of labeling reactions. Which I'll discuss in a few minutes, but also, you want to get good separation. And one of the separation, which was originally ring trials in the industry, was using this BEH column from Waters Corporation. And this is a small particle column, but it was used at high temperature and pressures in order to facilitate slightly fast separations. And we only injected about 1 microliter. So we ran the column at 60 degrees C. Again, this method was taken from an industrial ring trial, and the reference is given at the bottom of the slide. But you can see when you apply this type of methodology with the 2AB labeled N glycans, you can actually apply it to several different sort of structures. You can apply it to high manos, N glycans, fucosyl biotenary structures, N glycans, and fucosyl biotenary structured N glycans. And then based on the GU or glucose unit value from a reference calibration standard, you can identify the peaks. The peaks are normally identified by a library of glycans with their corresponding GU value. Again, all this information was taken from the same paper published in 2018 in Electrophoresis. So some of the ways that chemists and analytical chemists do to speed up this analysis is things like changing the particle size. And what I showed you previously was, say, a 1.7 micron particle. So it's a high pressure separation, or UPLC. This really replaces the historical slightly wider pore particle, a 3 micron particle, which had runtimes over double what that 60-minute runtime is shown. But if you want to go quicker, then what you normally have to do is really shorten the column. Shorten the column can impact the resolution a bit, but it really speeds up your runtime. So shorter column have been used. For example, the QTBH columns, again at 60 degrees C, will give you runtimes not 40 minutes, but down at 15 minutes. So you can cut it by a third because the column's now only 50 millimeters long compared to 150 millimeters. Again, this is all taken from the web, and you can pull this asset from this reference given at the bottom of the slide. The other thing you want to do to speed up sort of helic analysis is change your labeling reaction. And RapidFluor is a label which you can again buy actually from Waters Corporation. And this really speeds up the analysis because it speeds up the derivatization step. Where you were taking hours, now you take 35 minutes, and you can automate this. So this is, I would say, a way a lot of people now do a helic N Glycan analysis that uses RapidFluor reagent to label the glycans before they analyze them by HPLC. The one thing to bear in mind is this typically type of reagent typically requires SPE sample cleanup, so it can be slightly more expensive than other forms of derivatization because of the SPE required. But when you look at sensitivity comparisons, you can see them here, and you can see that they've moved from instant AB to RapidFluor MS labeling, and actually the relative performance improves. So actually there's some of this chemistries actually do improve analysis, okay, in fluorescence detection, but also, you know, as quite clearly shown on this slide, the RapidFluor is also MS amenable as well, and it tends to be better than the instant AB labeling. So a lot of people have moved to RapidFluor for helic type analysis. And again, this information was pulled from an application note which was written back in 2015. So why do people want to investigate and use capillary gel electrophoresis instead of helic for this analysis? Well, there are some benefits of using CGE or capillary gel electrophoresis. And for the next 5 or 10, 15 minutes, I'll go through some of these benefits. Now, the first thing to understand about CGE for N Glycan analysis, again, it's a charge-based separation technique, like a lot of capillary electrophoresis techniques. But again, you have to label your N Glycans because you need to be able to detect them. So we tend to label with a fluorochrome, so then they're picked up using laser-induced fluorescence detection. Typically, when you use CGE, you can either use coated or uncoated gel-filled capillaries. So we use, in this case, we don't use particles to do the separation, we use a gel. So we pass the sample, labeled N Glycan sample, through a gel, which then causes the separation in the applied field of the N Glycans. And there are several types of CGE solutions available in the market. It's a totally different separation mechanism to helic, but it's orthogonal. So it can give you additional information that you may miss sometimes with helic. But again, you use a similar method to identify the glycans in that you compare it to a GU reference standards or glycosyl unit reference standards, and that GU value you get from the CGE analysis will enable you to identify the glycans based on reference standards which already have a GU value. As I mentioned, in this case, these labeled glycans are detected by laser-induced fluorescence or LIFT detection when you're doing CGE analysis of N Glycans. Now, what we actually use in CE, the common reagent used for derivatizing N Glycans is 8-aminopyrene trisulfonate, or APTS, which is typically what everyone abbreviates it to. The really useful thing of actually using this derivatizing reagent is it adds a nice charge, negative charge, onto the glycan. So it allows you now, makes it polar, and it allows you now to make it move down the capillary when you apply a field. And so again, as with helic, you're moving from non-polar to a polar derivatized glycan, from a non-polar glycan, which enables you to separate it more easily in the separation medium, in this case CGE or capillary gel electrophoresis. So then looking at the historical prep time for this type of derivatization reaction, when you look at using APTS historically, it's pretty similar to the classical methods to sort of a 2AB derivatization, in that you have to, as usual, deglycosylate. So the first thing you do is you take your protein sample, as with helic, you strip off your N Glycans, you write down the sample, you label it with APTS. The labeling reaction typically takes 70 to 80 minutes in the classical kit. This is taken from Prozyme, again, instruction manual, similar reference I've already mentioned before. Then you clean up and remove the residual dry, and then the samples analysis. Typically, you're talking a couple of hours using for this classical derivatization method. Separation conditions, again, classically for CGE method, we classically used a coated capillary, N-choke coated capillary. This total length is 60 centimeters, but actually the length from injection to the detection window is a 50 centimeter effective length. And the internal diameter is a typo there, is 50 micrometers, not 50 millimeters. Capillary, the capillaries fill with a gel. It's the N-choke carbohydrate separation gel, which is a consumable you can buy from SCIEX. And we tend to fill the capillary by applying pressure to a vial or holding the gel for 3 minutes at 30 psi before each run. So effectively what you do is changing the separation meter every time you do analysis. Separation then happens by applying electric field across the capillary. In this case, it's 500 volts per centimeter at 20 degrees C for 20 minutes. Samples are injected by pressure. So you actually inject your sample by applying 2 psi for 10 seconds to the vial holding your sample. Again, this method was actually taken from a ring trial. This ring trial was actually mentioned in MABS in 2016. So again, all this information was taken from a peer-reviewed paper. But like HILIC, you can apply it to complicated glycan structures, high mannose, a fucosyl biantennary, and fucosyl biantennary. The example here matches how HILIC was applied. The key thing is highlighted in the red circle right at the bottom, is that the runtime with this classical separation is 20 minutes compared to the 40 minutes that was done on the HILIC ring trial paper in 2018. So actually CGE is actually quicker. However, as with using the rapid floor, we can also speed up the CE derivatization with APTS. And we've done this actually by using magnetic beads to improve the sample prep. Okay, so basically we add magnetic beads, we add our sample, we denature our protein, we do the PNG-ASF. The magnetic beads help bind onto the glycan and capture the glycans. We then use a magnet to capture the magnetic beads, which have the glycans. We then wash the beads to get rid of residual dye. Okay, we then label we first capture the glycans, we wash to get rid of some residual. We then label, then we remove the label. We then capture the magnetic, the glycans, label glycans again using magnetic beads, and then we elute off the glycans from the magnetic beads using water. This whole time, sort of derivatization time, takes roughly an hour if you look at the time, if you add all the times together on this slide. And then the sample's ready for CE LIF analysis, or CE capillary gel electrophoresis laser-induced fluorescence analysis. So as I mentioned, if you now put all these sort of methods into a table, you can see that the total runtime with this sort of cleanup is around about 60 minutes. So again, going through the steps in this particular fast glycan, as we call it, preparation workflow, you have magnetic bead addition, glycoprotein sample addition, you denature your glycoproteins, you cleave off your glycans, you capture magnetic beads, you wash it to remove the residual protein, and then you label it, you wash again to get rid of the excess dye, and then basically after you've washed the dye, you then release the glycans from the magnetic beads using water. We've also changed the method. As I mentioned, the historical method used a 60 centimeter capillary. As with HPLC, if you shorten the capillary, you speed up the analysis. So one of the things we did at SCIEX was we shortened the capillary to 30 centimeters. Now we've moved away from using coated to bare fused silica. It's just simple, more easier to handle. And now this capillary is half the length, so you speed up the runtime. The actual effective length separation here, again, is 20 centimeters because you basically have, out of 30 centimeters of the capillary, 20 centimeters gets you to the detection window. We use the same, we use a gel, again, exactly the same as the traditional method. So we again clean the capillary between each injection and add a fresh gel buffer. And we've increased the electric field, so it's like increasing the pressure. So you increase the speed of separation. So we increased it from 500 volts per centimeter to 1,000 volts per centimeter. And the actual runtime here has gone down from 20 minutes down to just under 10. The samples are injected again by pressure, as shown at the bottom of the slide. So now we've got really very fast analysis time. So we went from 20 minutes down to, this case, 5 minutes, as shown at the bottom in the red circle. And it shows you the separation of 25 different glycan structures. Now you'll see peaks labeled here DP2, DP3, DP5. What we do with CE is we add internal standards, which are GU reference standards, into the separation. We can then calibrate the GU value for all the other peaks using these reference standards, and it allows us to simply identify the peaks present in the sample, the glycans present in the sample, just in one run. We don't have to run an external calibration referenced ladder, which is often what you have to do in HILIC. So this really, again, is enables a fast turnaround of samples. And you can see on the table on the right-hand side of the slide some of the identified structures in this particular sample, which is run for a monoclonal antibody. Again, if you want to go back and slightly improve the resolution, you can always go back to the longer capillary, so go back to the 50 centimeter effective length, and you get slightly better resolution, as highlighted with the blue circle. But you know what you also do, you get better resolution, but you slow down your sample speed. So it's up to you on what you want to do, but it's quite flexible just by keeping the same method but just changing the capillary length. The other thing that was worked done actually by the team at Andres Gutman's team in over in Vesper, and they did a paper in this work in Hungary, the group I'm aligned to, and work with some. You can see that you can actually change the temperature. Okay, and here you can see an effective temperature, and you can beautiful thing you can do with our SCIEX system is the separation temperature can be controlled. So you can actually quite accurately control the temperature because of the liquid cooled capillary, and here you can see by adjusting the temperature we can get actually better separations. In this case, the separation between A and B, as shown here, actually improves when you run at slightly higher temperatures. So if you moved from, say, 20 to 30 degrees up to 45 degrees, you get a better separation of those two peaks. So again, if you got say co-migrating peaks, as we call in CE, maybe if you just play with the temperature, you can improve the separation you get off the capillary. Again, just comparing HILIC now with CGE, and this was data taken from an application. You can see here that this is work done in the US. You can see here that you can identify the same peaks with CGE as you can with UPLC fluorescence detection, but in a fraction of the runtime. So you know, even if you go with the fastest runtime, which is about 10-15 minute runtime with HILIC, and you compare it with the fastest runtime with CE, you are a lot faster because CE is off only about 5 minutes, where HILIC's 15, and you're still identifying the same peaks. Okay. The other thing you can now do recently in the last couple of years is you can actually now think about multiplexing the separation. And again, in the last year, SCIEX have released this type of assay now, which is available on the Biophase 8800 system. So now, rather than running one sample at a time, you can actually run 8 CE samples at the time. So you're running 8 runs of 5 minutes at the same time. So really your throughput then is a couple of minutes per sample. So it's dramatically faster than what you run on a HILIC with fluorescence detection. And again, you know, you always base this on comparing with a library, and you know, you get automatic peak identification with common N glycans, which are built into the library on this new system, which is the Biophase 8800. And the one thing we also do is we at SCIEX, we've actually worked with robotics companies. In this case, we work with Beckman Coulter, which is part of the Danaher family of companies, same as SCIEX, and we've partnered with them to develop a method to automate the sample prep for you. So you can actually put on the fast glycan sample prep, which uses the APTS labeling and magnetic beads, onto a biomech robot liquid handler, and that will actually prepare a plate for you. So you don't have to manually prepare the plates these days. You can actually apply a robot to do that. So you say you run in 8 samples with a separation time of 6 minutes. So really it's a couple of minutes per sample now with CE, compared to the fastest you could run is maybe 10-15 minutes per sample for on HILIC. So I'd just like to finish my talk with going through a quick summary, and this just summarizes some of the comments I've made today on how you could use CE to do N Glycan analysis. And really, I suppose, looking at the total layer, the latest modifications of UHPLC and CGE have dramatically reduced sample preparation analysis time. Both UHPLC and CGE methods require derivatization of the N glycans, but CGE uses a different derivatizing reagent. We use APTS rather than the rapid flow, which is, or A2AB, which is classically used for HILIC analysis. What we've actually done for both types of separation is there are now new methods to speed up sample prep. In the case of CGE, we use the fast glycan labeling kit, and this has really helped us to speed up the labeling process, and that can be robotized, put on a robot. And so when you compare the two techniques, really CGE is just faster. Okay, at the end of the day, you can run a sample a lot faster. And you can, again, if you want to improve the resolution of separation, as I mentioned, you can optimize the separation by adjusting the temperature, which is something you can do on SCIEX CE systems, because we have cooled capillaries. Liquid cooled capillaries, which allow you to really adjust the temperature and actually control the temperature separation. But for maximum throughput, I think something which is available on CE, which is not available by HILIC LC separation, is the ability to multiplex samples and to run 8 samples at one time on the new biophase system. That really gives us an analysis rate now of around about 1 minute per sample, so it's really fast. At that point now, I'd like to just finish my presentation for today, and again, I'll be open for questions and answers. I'll give you some answers at the end of this presentation. So again, we'll probably leave it to the end of the session where I'll happily answer any questions that people have listened today. And thank you very much for your attention.